Okay, hi class. This is chapter 17, Disease Resistance, War Within. And I would say that this chapter is sort of the general mechanisms of infection, how to transfer direct or indirect, and in later chapters we will talk about specific diseases, but this is sort of a general overview. Okay, so after reading this chapter, you should be able to discuss the characteristics and origin of the human microbiome, describe pathogenicity and distinguish between direct and indirect methods of disease transmission, distinguish exotoxins from endotoxins, discuss the first and second line immune defense against disease, describe how immune system requires specific pathogens. Compare the roles of cell-mediated immunity and antibody-mediated immunity. Distinguish active immunity, natural and artificial, from passive immunity, natural and artificial. Okay. The host and microbiome. The host and microbe, an intimate relationship. Okay. So the human microbiota basically means all the bacteria that are on you. And like any animal, you require a large number of different bacteria to sort of have your normal body functions. So in an infection, you have entry, establishment, and multiplication of a pathogen inside of the host. Um, infection is sort of the exception to the rule. It doesn't happen that often. Host, the organism which has the infection. Um, we tend to think of ourselves as hosts the most often because we mostly think of ourselves in terms of disease. Disease, this change from a good health state, and approximately 15 million deaths occur globally each year. Okay, and as you look at uh, that chart, there are a couple of those diseases where the deaths really aren't um, something that's a problem in the U.S. For example, the 1.8 million diarrhea disease, um, very, very few people in the U.S. will die of diarrhea. Um, we can generally get um, help for someone in this country. Um, the other thing, the tuberculosis and malaria are also two diseases that really don't cause much death in this country, but worldwide they can be a really huge problem. And actually, all three of those diseases are really dominated in poor countries. Okay. The human microbiome. So, the human microbiome uh, is the microbes that normally live on or inside of your body. Okay, and this is a huge area of research right now. Um, a lot of people are trying to figure out the microbiome, but we really haven't. Um, there is a service called uBiome where you can send off a sample, and for a price, they can sequence and tell you what bacteria you have. Um, I really want to do this, but I don't have the $100 to do it. and there are approximately 100 trillion microbes in a human body, um, which would have about 10 trillion cells. So one of the things that I think is sort of really amusing here is that you have so many microbes in your body, but you actually have fewer cells that are you. Okay, so really, we are just containers for carrying around those 100 trillion cells. Um, and I kind of think of it this way, that if some alien came down to Earth and was really looking at humans and the microbes, and all they could detect was life, not whether their detection system said it's human life or not, um, they would pretty much look right at your microbiome. They wouldn't look at you. They would sort of only care about your microbiome because it's so many more cells. Um, the microbiome totally out numbers the number of cells that you have. And this is an interesting chart, um, but most of the body tissues have some specific microbiome, microbes that live with them. Um, I'll give you one or a couple examples here, but um, one of the nice things is that the skin doesn't have that many microbes. Okay, um, Staphylococcus is the majority microbe on your skin, um, and the the staphylococcus that's there is really safe. Okay, it lives on your skin, but it's always there. You'll reduce the number of them when you take a shower, but pretty much they're going to be there all the time. Okay, that's one of the things that's great about the microbiome, is that it doesn't wash off. Um, Now, where you get into a really sort of ridiculous number of things is in your intestines. And there can be greater than 300 species uh, in your gut. Um, and when you do, say, digestion of a food, it's a combination of all the microbes together. There's not just one microbe that goes with one kind of food or something like that. Um, you're you're talking about a community that's going to do the work of doing the digestion. Um, so the human microbe is mostly a symbiosis. So in symbiosis, that means that the bacteria and the human are living together, usually in harmony. Okay, both sides get a little bit. Um, if it's mutualism, then both parties are going to benefit. And you can think of that something like when you eat some food, okay, the microbe benefits because you have given them food to digest, and then the human benefits from the products of the digestion. And in communalism, that means only one of the two benefits, um, and not that common in the microbiome, but maybe you could envision a scenario where, say, the someone in your microbiome has a antibiotic resistance, and uh, if you take the antibiotic, you'd be benefiting that one member of the microbiome that's resistant. And so it's very important to maintain your proper microbiome. The biggest thing for that is diet. Okay, and you can also take probiotics. Um, Probiotics, uh, generally the first one that comes to mind is Lactobacillus acidophilus, um, which is the lactobacillus is the first bacteria that ever colonizes your gut when you're a baby. Okay, inside the womb, the baby is sterile, and then as the baby passes through the birth canal, it will pick up microbes from the mother. Um, those microbes that are on the mother again get ingested by the baby, and that provides um, the start of the microbiome. Um, with probiotics, there you're taking a supplemental bacteria to try to boost up your microbiome. And like I said, lactobacillus, which again is the first bacteria that is ever colonized, it's a very popular probiotic. Um, there are certain kinds of yogurt that will say they have probiotics in them, and they don't really say what bacteria they have in there because it's patented and um, it would be against the company. Um, but I think that any of those things that uh, say they have probiotics in them, um, some are probably better than others, but in general, I think taking a probiotic is a really good thing. Okay, so as I was just saying, um, at birth, a newborn is essentially sterile. Okay, certainly within the womb, the baby is sterile. Okay, and the colonization can come from a couple of different sources, but like I said, the very first place you're going to get some is passing through the birth canal. Okay, then things like feeding, um, just being touched by doctors or other people, um, breastfeeding, um, anything that is touching other humans is going to cause some of the microbiome to spread. Okay, and I don't think that it's been done yet, but I think it'd be very interesting to see uh, the exact species of, say, the Staphylococcus that are on um, your mother, your father, and then the newborn, and see how closely related, or maybe who was the first person to touch the baby, because that the baby would have the microbiome of the person who first touched the baby. Okay, so we can have microbiome differences if you have a natural birth. Again, if you do a cesarean section, then the baby's not passing through the birth canal. So where do you get the microbiome? Because normally we get it from passing through the birth canal. And you do have some very dynamic situations with the microbiome, and they usually sort of settles down by about age 3. And when you get older, you can start losing some of your microbiome, and so people who are quite elderly, say maybe 85 or over, it's probably really important for them to take um, probiotics so that they can keep whatever microbiome that they have. Okay, like I said before, this chapter is a lot about general mechanisms of disease, not specific diseases, but just how the disease process works. So now we'll talk about some of the concepts of infectious disease. Okay. Pathogenicity versus virulence. Okay. Pathogenicity means the ability to enter the host cell and cause disease. Opportunistic pathogens are those that can do the entry into the host and cause damage when the host is slightly compromised. So that could be dietary, that could be tiredness, um, it could be a bigger problem like a weakened immune system, or your immune system temporarily shuts down a little bit. But all those things that are a problem with the host give an opportunistic pathogen the ability to go on and cause disease. Okay. Now, virulence refers to the level of damage done by the pathogen. Okay. Um, so, if something is avirulent, sorry. Okay, those terms were basically on the individual level. Now let's talk about large population terms. So we have the terms endemic, epidemic, and pandemic. So if something is endemic, okay, that means it occurs at a low level in a population in a certain area, okay? And so we have a number of diseases that are endemic, and you sort of always have the chance of getting those endemic diseases because there's someone in the population that has it, but it's at a very low level. Now, in epidemic, you have an explosive increase in the cases of the disease. Okay, and we think about epidemics as being so scary. Um, One of the things that sort of has been sort of a new epidemic, or at least a recurring, but we get epidemics of flu fairly often. Okay, and so we get swine flu or some other form of flu that you just almost everyone gets it. Um, Now, when you have an outbreak, then you're going to be more confined than an epidemic. Um, so, for example, if you had disease that occurred in all the populations of uh, Galloway, that would be an epidemic. But if we only had people who were getting uh, sick on campus, that would be an outbreak on campus. Now, when you have an epidemic that spreads worldwide, that's what's referred to as a pandemic. Um, so, and this definitely occurs. Um, it sometimes takes time to spread the disease, but depending on which disease it is, you can get worldwide spread fairly easily. So something that's airborne, like flu, for example, um, not that hard to transfer worldwide because the it's so easy to catch things that are airborne. modes of transmission. So we can have direct contact, and this seems pretty obvious, but if you touch the person, um, so person to person, like holding hands, um, or touching the person somewhere, that will be your um, uh, human to human direct contact. Um, from respiratory droplets, when someone coughs, this is why it's always important to cover your mouth when you cough so that you don't spread those droplets. Um, or you could have a direct contact by petting your animal. Um, Indirect methods, uh, something like contaminated food, contaminated water, or using some object that's contaminated. So you can see there the person stepping on a nail out in the yard, and that is contaminated with the soil bacteria. Um, Using things like Q-tips or putting contacts in, those could be contact with that inanimate object. Okay. We can also have arthropod spider bites, which are going to cause a whole lot of damage. Um, of these indirect methods, um, contamination of water and contaminated food are probably the two biggest spreads. Okay. Um, Now, water is a problem because obviously, if the water is contaminated, lots of people can drink from that same source. Um, and that's why we have so much good water decontamination in this country. We talked about that last chapter on how you actually do that purification system. And like I said then, it's really important that in this country we do have really good water conditions. So in terms again of transmission of diseases, you can have something that's a reservoir. So in a reservoir, this is where the pathogen is going to reside and multiply. Okay, and the reservoirs don't have to be animate objects; they can be inanimate objects, um, like something. Let's say you put a open container of yogurt in the refrigerator. Um, that yogurt could become a food source, and if it got contaminated while you were eating it, um, that could mean that the pathogen could reside in there and multiply, uh, even though it multiplies slower at cold temperature, but still that container of yogurt could then be a reservoir. Now, zoonosis, okay, zoonosis is when the reservoir is a animal. Okay, so there definitely are animals that can take, can live with a one of the pathogens living inside of them, because say, and this especially happens with viruses, uh, but because viruses are very specific to being human viruses or animal viruses, um, you could maybe have the virus inside of an animal and it could multiply, but it's not uh, giving a disease because that virus is human specifically. Okay. Um, so we can look at this summary, um, and you can see that you can have a population that are susceptible hosts, hosts. You can have uh, a mode of transmission that is passing to the uh, susceptible host, and you can also have a source of a pathogen. And when those three things get together, then you get the infection. Okay, so uh, you know, when we start talking diseases, people start to think that everything is going to cause disease, but you can see that those three things have to come together to cause the disease. So uh, being being infected is sort of the minority player. Um, the microbes don't want to make you sick. It's just something that happens when you get uh, a susceptible host, a good source, and a good mode of transmission. Okay. So as the disease progresses, um, we first have signs. Okay. Um, so you get a sign that you have the disease, and this is generally predicted or noticed by the doctor, so you have some sort of evidence that the disease is there and it's detected by the observer. Uh, Symptoms are evidence that a disease is sensed by the patient, and syndromes are specific groups of signs and symptoms. So if you have very reproducible symptoms, then that might be becoming a syndrome. Now, in terms of stages of the disease, um, you can have the entrance of flu virus, for example, on about the first day, uh, then from about the fourth day to the 10th day or so, um, then you get the all the symptoms, okay, and then after about the 11th day, you're in your convalescence period where the symptoms are going to uh, start to terminate. Okay. Now, I want to point out though that see that line uh, says infectious period. Um, those are the times when you can spread the disease to other people. Okay, so you might say, "Oh, well, I don't have that many symptoms right now," um, but you could very easily still spread the flu um, just because uh, you have the virus present. The virus will start to multiply, and you breathe it out, and so you're spreading the disease. Um, and so sometimes people don't think, oh, well, I only have one symptom right now. I can't be that infectious, or I'm not all that sick. I'm not going to spread the disease to other people. Um, you really can't. Um, the other thing on the other side of that, sometimes people don't give themselves enough time to get better. Okay, we all have things we need to do, um, and so even though we're very, very sick, fever or something, um, you may want to not go back to school or go out and do things um, because you need to recover. Okay, um, and I think it's sort of a good rule of thumb. Um, After you have, say, a high fever, when you're feeling really sick, um, take the next day off too. Okay, don't push yourself because again that might be one of those uh, things that are going to make you susceptible again to a new disease. Okay, establishment of disease. Um, so we have to think about how the bacteria or virus is getting into the cell, and so we have to worry about entry and then invasion. So the first thing is the portal of entry. And the portal of entry, again, we have to worry about body sites. Um, Different portals of entry are going to matter. Um, a sort of easy portal of entry is obviously your mouth, and so you can ingest food very easily. Um, if you have a break in your skin, so you have some sore on your skin, that could be a portal of entry. Then, in terms of invasion, we have invasiveness, which is the penetration or structural damage of the cell. And as you look at the picture on the bottom, um, you can see that we have the microbe and we get to a portal of entry. So we have some cell that has been opened up, and then that cell is going to be susceptible, and the bacteria are going to multiply inside of it and do its damage. And then we also have the portal of entry, where those multiplied bacteria are going to exit that cell. So bacterial toxins. So I always find this a kind of exciting topic. Um, we have two types of bacterial toxins, exotoxins and endotoxins. Now, the exotoxins are heat-stable proteins and trigger a strong immune response. Okay, so you can have antibodies produced against the exotoxins. Um, the really important thing here, or important things, are that the exotoxins are proteins, and that those proteins are very heat stable. Okay, um, uh, heat liable. They break down in heat pretty quickly. Okay. Um, Botulism and cholera toxin are two very good examples of that. Botulism toxin is probably the most dangerous toxin that is known to man. Okay, which is really sort of amusing how people do Botox injections to try to do skin things, because if they screw that up, you will become paralyzed. And just since I'm sort of talking about that, um, when you have botulism, you die uh, uh, of becoming paralyzed. Okay, and what usually happens is your diaphragm becomes paralyzed, and then you can't breathe anymore. So this is a problem again with Botox. If the Botox injections are done not appropriately, the doctor could give you too much Botox and could cause some paralysis. Uh, a former student of mine did have that problem. She got uh, Botox injections in her forehead and eyes, and basically her entire face became paralyzed. And it lasts quite a while. It's probably a month or so that she had her face paralyzed. Now, a toxoid um, can be used. It's a part of the toxin, part of that protein, and that can be used in vaccines. Okay, and there are a number of vaccines that use this kind of system where you inject a small part of the toxin, um, and uh, then your body will make antibodies against that small part of the toxin, which should hopefully give you immunity to that toxin, the whole toxin. Okay. Um, when you have that toxin, you generally sort of cut off maybe an arm of the protein so that it's not the full protein anymore, and it's just a part of the protein, so that it's better to make an immune response, but it's not going to cause disease. Okay. Um, Endotoxins are heat stable, um, and they are part of the cell wall of um, the bacteria, um, and they don't trigger a strong immune response. Okay. Now the endotoxins are not as dangerous. Um, they are sort of very normal to have the endotoxins around. Um, Typhoid fever is an example of that. Um, Several of the food poisonings are also examples where the endotoxin, while the food might not have that super dangerous protein of the exotoxins, just because the bacteria are present, they'll have the cell walls, and then that makes those endotoxins. Um, I mentioned it before, but again, remember that after the infection process happens, we have to have a portal of exit. Okay, so the pathogen needs to exit from the cells, um, and that's going to be cell type specific, how the cell is going to decide to get rid of that pathogen. In some cases, the cell might just die. In other systems, there might be a organized pump to pump the pathogen out of the human cell. Okay, we can have a number of different resistance mechanisms. The first type of nonspecific resistance disease we'll talk about are natural born immunity. And in natural born uh, immunity, you can kind of think of this as an umbrella of protection. Okay, so that if you have a pathogen, okay, um, then you can have other microorganisms or something that can give you sort of this protection under here. Um, so again, non-specific ways that we can use this umbrella to keep the pathogen from actually entering into our body. Okay, because like we just said, you have to have an entry um, besides transmitting the disease to you. You have to have a way of having entry, and then once the entry happens, then you can have some non-specific mechanisms that are going to prevent the pathogens from establishing itself inside of the body. So there are a number of mechanical and chemical barriers. Your skin is a very good method for keeping things from entering. Um, Now, you do have cells sloughing off, and so, you know, if the pathogen lands on your skin, then it there are cells on the top layer of your skin that are constantly being sloughed off, and that can take the pathogen with it, so that you don't get entry into the body. Um, a good example of that would be, say, when you shower. Okay, because you lose a lot of skin cells when you shower, and so if the pathogen was on you, you take a shower, that um, takes the skin off, and taking the skin off will take some of the pathogen with it. Um, Now, your skin in a deeper layer has keratin. You also have sebum, and that is the uh, sweat gland production, and that's going to keep you uh, from getting diseases on the skin. Okay. The skin is definitely a portal of entry. Um, and again, usually you have to damage the skin for it to be a portal of entry. Okay. Um, Mucous membranes. Um, think about that. Um, say your mouth or your nose, around your eyes, you have a mucus, and that mucus is going to trap the microbes. And in general, that trapping is going to be sloughed off again. Um, so think about that with your nose. Um, if you breathe in a pathogen through your nose, it's probably going to get trapped in the mucous membranes. And then when you blow your nose, you can get rid of that mucus and then get rid of the microbes. Okay. Um, Same thing with your eyes. You might not think that much about crying, but let's say that just from the air, your eyes get a pathogen in them. The pathogen can get trapped in the mucus of your eyes, and then as you cry, you're able to flush those membranes so that it flushes out all the pathogens. Now we can have chemical barriers, and this is mostly done through pH. Um, and Vaginal pH and stomach pH are fairly low, and that will allow only a very, very few microbes to be able to grow there. Okay, it's very hard to get a stomach infection because the pH is so low. Um, the one that gives you ulcers, Helicobacter pylori, has mechanisms to be able to deal with such low pH. In your eyes, there's also an enzyme called lysozyme, and lysozyme actually breaks down the cell walls. And we use lysozyme in my lab all the time. If we're going to extract DNA from a cell, um, one of the first steps is to add some lysozyme, because then you're breaking down all the cell walls and you can actually get to the DNA. Okay. In terms of viruses, interferon will protect cells, and virus infections can't really multiply when interferon is present. Okay. Um, so inside of a cell, you can have a process called phagocytosis, and in phagocytosis, you are ingesting the microbe. Okay, so here we have a bacteria and this macrophage, which is one of your kinds of white blood cells, um, that can sort of non-specifically take up that bacteria through the process of phagocytosis, and then now that the phagocytosis has occurred, that bacteria is actually inside of the red blood cells, and it's going to interact with these things called lysosomes. Okay, and in those lysosomes, there are enzymes which are going to kill the bacteria. Okay, so here the lysosomes have started going around that bacteria, and then they get digested, and you get a uh, vessel inside of the white blood cell that has all the garbage that was the bacteria, and then uh, we can do sort of a reverse phagocytosis and eject all those little particles which used to be the cell, but are now just sort of waste product. Um, Signs that you have an infection. A key sign of infection and a nonspecific resistance mechanism is inflammation. Okay, so in inflammation is a response of a tissue to a traumatic event. You can have inflammation from both a disease process or from physically hurting an object. So you fall down and your knees will have an inflammation because uh, you put trauma onto your knees because you fell down. Okay, so signs of inflammation: redness, warmth, swelling, and pain. Okay, and that doesn't mean that you have to have all those to be inflamed. If you only have, say, redness and swelling, uh, that is still inflammation. Okay. Um, like you're saying, sebum is the sweat glands and what's being produced by them. Okay, and at the inflammation site, this can often be a portal of entry because again we have some uh, tissue damage. Okay, so when you have that cut or abrasion, and especially if there's uh, even more inflammation than just a cut, that's going to serve as a great portal of entry. Now, another non-specific thing is fever. Okay, and fever, of course, is a rise in the body temperature, and that is caused by pyrogens. Okay, um, and the pyrogens are a class of organisms which, when they're in the body, they trigger the body to raise the temperature. Um, these can be immune proteins or microbial fragments which are causing the fever, can serve as pathogens. Okay. And as the pyrogen is going through the body, it's going to trigger the hypothalamus to increase the body temperature. Okay, we just went over some of the non-specific resistance things like fever. Now we're going to talk about specific resistance, and this is where you have your acquired immunity. Okay, so um, we have to be able to recognize the pathogen, and to do that, you have an antigen. So an antigen is part of a molecule that is recognized by T cells, B cells, and antibodies. Okay? And the number of cell types in the immune system starts getting really, really high pretty quick. Okay, so you can see we have now T cells and B cells, and those are used to produce antibodies. Okay. And the antigen is usually a protein, sometimes carbohydrates or synthetic substances can be the thing that triggers the T cells or B cells. Um, Now, when we talk about uh, what the antigen is, this could be a toxin, or the flagella or pili, or the viral capsid. Again, those are all proteins, and they're all very specific proteins. Um, so they're they're something that's going to be recognized by those cell types and produce antibodies to it. Now, the epitope is the part of the antigen that is recognized. Okay, um, so let's say you have a toxin. Uh, the entire toxin could be the antigen, but only a little tiny part of it is going to be the epitope. Um, the body is going to be able to recognize whether it's self or non-self epitope. Okay, and this is very important because you want uh, the foreign object, the pathogen, to be recognized, to have epitopes that are only recognized by those foreign things, and you don't want your own cells to be attacked. Um, the if the epitope is not specific enough, it could start attacking human cells instead of just the pathogen. Um, in terms of the cells of the immune systems, we have lymphocytes, okay, and your B lymphocytes or your B cells, and you have your T cells, uh, which are the T lymphocytes. So, like I was saying on the previous slide about B cells and T cells, um, these are all types of lymphocytes. And your cells, your lymphocytes, are made inside the bone marrow. Okay, and then those lymphoid stem cells are going to go on to become either B cells or T cells, and then those secondary lymphocytes are going to be things that are going to go on to uh, find antigens and be able to move on to doing cell-mediated immunity or antibody immunity. Okay, so your B cells are going to produce the antibodies, whereas your T cells are going to go on to kill your other cells. Um, so there's like let's say a cell is infected with the virus, then you'll probably want a T cell to be able to go and kill that cell that's been infected. Um, and while circulating in the blood, you probably want some of those B cells so that antibodies can be made and attack that virus. Okay, steps in this process. Okay, so the pathogen has to be recognized. and then we have the T cell or control activation, and finally control factor release, and that will end up in the death of that cell type. Now, an antibody response, again, we have a cell type, a macrophage, which is going to be able to recognize that something is an antigen, and then with the help of the T cells, we'll get these activated helper T cells, and they will then go to a B cell with the antigen that is being presented to it with the help of the helper T cell, and then the B cell will then go on to make more B cells with the antibody present, and a few of those cells go into memory cells, and the rest of those are plasma cells. The memory cells sort of hang out in the body, and the antibodies are being formed by these plasma cells. Now, one of the great things about these memory cells is, like I said, they're going to go hang out in the body, and so you can't really get the same disease twice as long as you still have good memory cells. Okay, and that's why, so say for a common cold where you're excreting a lot of the virus from the cold, you're not going to get the same strain of the virus right away because you have those memory cells which are giving you memory. Now, those memory cells, especially for something like the cold, don't last very long. Okay, so you can get a cold the next season or maybe even the next month because your memory cells aren't that popular, and or they haven't lasted that long. Um, the other thing it could be is you could be getting a cold from a different virus that has different epitopes. Okay, so you can only develop the memory cells to the epitopes that were presented in that cell. Structure of an antibody. Um, so the basic structure is that you have light chains and heavy chains. Um, so the sort of more technical way for to say antibody is an immunoglobulin, or Ig protein. Okay, and that's illustrated in that figure there. Okay, so we have two light chains. Um, these little chains are the light chains, and then we have two heavy chains, which are those very long uh, proteins. Okay, and I'll point out that see how at the end of the heavy chain light chain complex on those arms, where the antigen is binding, those are highly variable. Okay, so if you were just to extract immunoglobulins from a human, you would find that, say, the tail region is all going to be very consistent. Okay, it won't change the structure at all. However, uh, at those antigen binding sites, there's going to be a different uh, antigen binding site for every kind of pathogen that has entered that cell, for every epitope, actually, that's in that cell. Okay, so we have a couple different classes of antibodies. The IgG, which we just talked about. Okay. We can have IgM, IgA, IgD, and just let me one more, an IgE. Let me point out here that the structure is sort of always the same. You always have the two heavy chains, two light chains, and then at the ends of that light chain, heavy chain, you have the binding site for the antigen. And the IgGs are the ones that are floating free in your system. Um, things like IgM and IgA are uh, for certain kinds of immunity. They're more specific to what they do. Um, IgE can be used in when you have allergies to things. Types of immunity. Um, Again, we have our natural immunity, um, which we're being exposed to antigen um, and often following a disease process. So you get, um, you know, a response from having the disease. You get some additional immunity because that pathogen has been in your body. Okay. Um, in panel B, um, we can have a artificially acquired um, through a vaccination. Okay, so in that case, we are giving a bit of the virus or an epitope of the virus so that our immune system will recognize that uh, to do an active immunity. Now, in passive immunity, um, here, like the baby um, is obtaining antibodies from the mother. Okay, and so those are generally the IgG, and it's going to pass across the placenta, and then the baby's going to have some inborn immunity because it has taken some of the antibodies from the mother. The other way to do artificially acquired is to transfer antibodies from a person or animal that has been given the antigen. And this is something, and it's sort of generally done in very large animals. So you can give an animal part of the toxin and then take the blood from that, let's say cow, and then so bleed the cow and take its blood and put it in with your blood, and then the antibodies will be inside the cow's blood, which will attack that virus. Okay, so what we should have learned from this lesson: the intimate role of the human microbiome in the maintenance of good health, characteristics of infectious diseases, including virulence, disease within a population, disease transmission, and the course of a disease. how bacteria can use disease, entry, the toxin production, and the exit, defense of the natural born immunity, acquiring of cell and antibody immunity, and types of specific immunity. Okay. And use topics of this chapter. How would the human body attempt to inhibit the infection process by a flu virus? How does a flu vaccine mimic this process? So, I've mentioned flu several times during this, but just think about each of the steps. How would a flu virus, go on to give the flu? And what things does a flu virus have to avoid so that the immunity doesn't get that virus? All right, thank you.